All right, so now that we've already discussed the lipids and the carbohydrates that are within the plasma membrane or make up the plasma membrane, let's turn our attention now to the proteins that are associated with the plasma membrane. So recall that peripheral proteins sit on either the inner or the outer surface of the plasma membrane, and integral proteins are actually embedded within the plasma membrane itself. So the integral proteins may actually span the entire width of the membrane, or they might just be embedded on one side of the membrane. So let's look at function. So what is this variety of proteins that in the plasma membrane? What does this variety of proteins do for our cells? And it turns out these proteins do a lot for our cells. So they have many different functions, as you can see here. We'll highlight each of these and then real briefly here, and then we'll explore each of them in a little bit more detail in a few moments here. So some membrane proteins function as channels or carriers that transport molecules across the membrane. So they aid in the movement of materials either into or out of the cell. Some function as receptors that bind hormone or neurotransmitters. So this binding of the chemical messenger to the receptor or the receptor protein causes a biochemical change within the cell. And this biochemical change is referred to as signal transduction. And it's these changes that alter what the cell is doing in response to that particular chemical messenger. Some of these membrane proteins function as enzymes. So recall that an enzyme is responsible for either building or breaking down substances. So as enzymes are biological catalysts. Some function as attachment points to a structure called the cytoskeleton that maintains cell shape. These attachment proteins also assist in movement of the cell. So the cytoskeleton is actually a network of protein fibers and filaments that's within the inside of the cell that supports the cell from within. So much like our skeleton supports our body from within, the cytoskeleton is not bone or bony material, but it is a network of protein fibers and filaments. And other membrane proteins function as cell-cell junctions, which permit the cell to adhere and stick to other nearby cells. So let's take a look, let's expand more upon the, starting at the channels and carriers function of membrane proteins. Let's expand upon that for a minute here. So as we described in the previous slide, we saw that some membrane proteins function as channels or carriers to allow the movement of materials across the plasma membrane and into or out of the cell. So membrane protein channels that function in transport tend to be water-filled, so that is, they really just behave like a small water-filled tunnel or passageway across the plasma membrane. So recall that the core of the plasma membrane was hydrophobic, and the lipid tails that occupy the core of the plasma membrane don't easily allow water to pass from one side or through that lipid core to the other side of the membrane. And so these water-filled transport channels provide that passageway, that water-filled passageway across the lipid part of the membrane. So these channels allow for the passage of hydrophilic ions. So there's that term hydrophilic, which means water-loving. And so these hydrophilic ions are things like sodium, potassium, calcium, might even be glucose. So since glucose is a hydrophilic substance as well. So this allows for the passage of water-loving substances across the plasma membrane. We get two flavors or two varieties of transport channels. We have what are called leak channels, and another variety, they're called gated channels. So leak channels are channels that are always open. That is, they allow the movement of substances across the plasma membrane at any time. So that is, there is nothing regulating the opening of this membrane protein, and so that allows for the movement of these hydrophilic substances at any time without any hindrance there. Now, gated channels, on the other hand, are normally closed, and they typically open in the presence of a stimulus. And so in this example here, we have our gated membrane protein channel. And in this example here, our stimulus is a chemical signal. So for example, a hormone or a neurotransmitter, that will be our chemical signal. So normally, without that chemical signal present, or without that chemical signal bound to our membrane, our gated membrane channel, that channel is closed. So you can kind of think of this as like a closed doorway. But when that stimulus binds to our channel, our gated channel here, that changes the shape of our channel, and it opens it up and allows the passage of these two ions across the membrane here. So in this way, the chemical is kind of acting like a key to open up the gated channel doorway across the plasma membrane. So in this example, the stimulus was a chemical stimulus that actually had to bind to our membrane channel and open it up. But the stimulus can also be a change in membrane voltage, or a change in voltage across the membrane. So it's kind of like a little electronically operated door, if you will. All right, so we've explored two different types of channels. We've seen the leak channels and the gated channels. So why are these proteins important, or why are these channel proteins particularly important? So of course we discussed the fact that it allows for passage of materials into and out of the cell, but what purpose does that serve? Why do we need materials to move into and out of the cell? Right, first of all, the cell has to be able to use new materials to construct things. The cell also has to be able to get rid of wastes and eliminate things that it doesn't need, and so these things have to be able to get across the plasma membrane so that the cell can have its raw materials to build things, but then also eliminate the wastes that it does not need. The channel proteins are also highly important in maintaining the intracellular levels of two ions, in particular potassium and sodium. So K+ is our potassium ions and Na+ is our sodium ions. And so channel proteins are also highly involved in regulating the amount of sodium and potassium in our cell during resting membrane potential. So we'll unpack this term in just a second here. So when the cell is at rest, that is, when it is not stimulated, that's what we refer to as resting membrane potential. And at resting membrane potential, there is a high concentration of potassium maintained on the inside of the cell, with very low sodium concentrations inside of the cell. Now, the opposite is true outside of the cell. So outside of the cell at rest, there tends to be very high concentrations of sodium and very low concentrations of potassium. So resting membrane potential is a characteristic of most of the cells in our body, and it's particularly important in cells like neurons and skeletal muscle cells, as this resting membrane potential changes when the cell is stimulated. And we see a number of biochemical changes that happen when resting membrane potential changes. And then those biochemical changes lead to the signaling in neurons and lead to the contraction in skeletal muscle cells. So hopefully I've highlighted the importance of channel proteins and why they're very critical in the normal functioning and operation of cells. But what we also see is that defects in channel proteins cause disease, and in some cases very deadly diseases. And so, for example, a disease called cystic fibrosis is actually a disease that's caused by a defective chloride channel protein. And that chloride channel protein is called CFTR. Now, a normal functioning CFTR channel, which is here on the left, moves chloride ions from the inside of the cell to the outside of the cell. And water follows these chloride ions passively with it. And so what these chloride ions and water, what this mixture of chloride ions and water does to the outside of our cell is it creates a thin layer of saline or salt water on the surface of the cell. And this is particularly important in the lungs and in the respiratory system. However, a defective or a mutant CFTR channel, which is shown here on the right, is unable to move chloride ions to the outside of the cell. And so this means that there is a lack of saline water on the outside of the cell, which causes the buildup of mucus and debris on the surface of these cells, and this buildup of mucus and debris often leads to frequent and chronic infections. And so that's what you see with cystic fibrosis, is that you see often persistent and chronic infections of the respiratory system. You get fluid buildup and mucus buildup, and generally it just kind of makes breathing and then other things hard, or much harder for the person who has cystic fibrosis. Now, cystic fibrosis is a disease that that you get when you are, so it's a disease that you acquire before you are born, and so cystic fibrosis is not a disease that you can contract or that you can pass from person to person. So you have to be born with it, as the as your DNA and your genetics determine whether your body will make the normal CFTR channels or the mutant defective CFTR channels. So other membrane proteins function as receptors for chemical messengers, and so in this example here, a chemical messenger, so like a hormone, will often bind to a protein receptor that's on the surface of our cell, the plasma membrane here, which is abbreviated PM. So this receptor protein then activates a G protein, which is, this G protein is typically located on the inside of the cell. And this G protein then goes on to activate a second messenger inside of the cell. And that second messenger then changes the behavior or the response of the cell as a result of that chemical messenger binding to the receptor protein. And so this setup, or this configuration, is necessary because many chemical messengers actually cannot enter the cell. That is, they cannot cross the plasma membrane. They don't have the ability to cross the plasma membrane and change the cell's behavior. But thanks to this chemical messenger system here, or this second messenger system here, and we'll unpack this term second messenger in just a second here, but thanks to this receptor protein, these chemical messengers, even though they can't get across the plasma membrane, these receptor proteins and these G proteins still have an effect on, or these activated G proteins still have an effect on what the cell is doing, so it can change the cell behavior. So as I mentioned, this type of messaging system, where we have a chemical messenger activating a G protein and then that G protein creating a second messenger, is referred to as a as a second messenger system. And so here's another kind of a little bit more simplified diagram showing the second messenger system. So we have the outside of the cell towards the top, the plasma membrane here in these little black ovals with their lipid tails, and then the inside of the cell down here in blue. We have our chemical messenger here, so something like a hormone will often bind to a specific site or a specific shape in the membrane protein. So this is our membrane protein receptor here, which is very specific for this chemical messenger right here. So when this chemical messenger binds to its membrane receptor, that membrane receptor is associated with an inactive G protein. When this chemical messenger binds to it, that G protein will become active. That activated G protein will then stimulate another protein within the cell that's usually called an effector protein. That effector protein then creates a second messenger chemical or a second messenger molecule. And that second messenger molecule then changes the behavior or influences the cell's response to this initial chemical. So this is called a second messenger system because this chemical that's made is the second messenger in the pathway here, whereas the hormone or neurotransmitter chemical was the first messenger in the pathway here. So some membrane proteins also function as enzymes. So recall that enzymes are biological catalysts, which take two or more substances and combine them together to make a new substance. Or they break substances down into smaller components, which is essentially kind of what this diagram is showing here. It's showing this molecule here being changed into something else, which interacts with after it interacts with an enzyme. Then this molecule right here interacts with another enzyme and changes it again to something else. And so a little bit, even though this diagram is a little unclear as to what it's showing, basically it's showing that enzymes are catalyzing the formation, or biological catalysts that are creating or breaking down substances. Finally, or I should say last but not least, we have certain plasma membrane proteins that function as cell-cell junctions. Now, these junctions join the plasma membranes of nearby cells to one another, so these are essentially linking cells together in different ways. As you can see from this list here, there are 3 types, and each of these types of cellular junctions has a specific function. So we have desmosomes, we have tight junctions, and then we also have gap junctions as well. So let's take a look at what each of these does. So desmosomes tend to function a little bit like rivets that anchor nearby cells to one another. Essentially what a desmosome is it's a number of protein plaques, fibers, and filaments here. And what you have in desmosomes is that you actually have these small little linker proteins that extend from one cell to another and link them physically together or bind them physically together. So what you're seeing in this diagram here are 2 adjacent cells, one on the left, one on the right. This is the plasma membrane of one of those cells. This is the plasma membrane of the other cell. And then in between you have these little teeth-like proteins that extend out from the surface of each of the cells and link together and interlock like teeth in a zipper. So in tissues that are linked together by desmosomes, they behave essentially as one continuous sheet, and so that makes the tissue really highly resistant to what are called tensile forces. Tensile forces are simply referred to as pulling forces. So these cells, or this tissue that are that's linked, the cells of this tissue that are linked together by desmosomes is really resistant to tearing. And so as you can imagine, desmosomes are particularly abundant in really tear-resistant tissues, and so things like the skin and cardiac muscle are both tissues that are subject to really strong mechanical forces. And so it's important to link those two, to link the cells together in those tissues together so they do not easily become separated or torn from one another. Now, tight junctions, on the other hand, function to seal the space between adjacent cells. So this is kind of this could be compared to the way that a Ziploc or a resealable bag essentially kind of snaps together. And so you're sealing the space so that fluid and materials cannot move between between adjacent cells where the seal is formed. So this prevents molecules, fluids, ions, materials, things like that from getting between the cells and getting into deeper tissues or things like that. So therefore, the cells that are linked together by tight junctions can function to essentially separate one fluid compartment from another fluid compartment in the body. In kind of much the same way that the plasma membrane separates the fluid compartments between the inside and the outside of the cell. So this type of cell-cell junction is highly beneficial in the epithelial tissues of things like the digestive system, as well as the skin. So in the digestive system in particular, tight junctions prevent digestive acids or digestive enzymes from seeping between the lining of the stomach or the lining of the intestines and getting into deeper tissues where they could cause some damage. And finally here, gap junctions function as communication channels between adjacent cells. So nearby cells are essentially connected. Gap junctions actually connect nearby cells together because gap junctions essentially kind of resemble these hollow little tube-like proteins. And these hollow little tube-like proteins allow fluids, ions, chemical signals, and things like that to pass from the interior of one cell to the interior of another cell. So superficially, they might look a little bit like desmosomes, or maybe even a little bit like tight junctions, but these are actually small little channels or tunnels that are connecting the interior of one cell to the interior of an adjacent cell. So these gap junctions tend to be present in electrically excitable tissues, so things like cardiac muscle, where electrical signals can more easily spread from one cell to the next. So that way the heart can contract, or the heart muscle, I should say, can contract simultaneously and without delay. But they're also found in embryonic tissues, because it's important to, in developing tissues, since the cardiovascular system may or may not be fully developed yet, or I should say may not be fully developed yet, it's important to allow materials to pass from one cell to another in developing tissues, so that way those cells have all the materials that they need in order to function correctly and build the things that they need.